Let me introduce Gabriel Kreiman. This is the best introduction I can come up with. He said it's correct. He found Bill Clinton cells and then he came to MIT. Now he's at Harvard.
Thank you very much for that very candid and long introduction. I'm in the CRC mailing list. I always mean to interact more with this crowd. I'm very interested in this set of questions, and life happens, and I haven't been able to interact with this group as much. But I really was hoping that this was going to be informal and interactive, and people really ask me questions. Then some people said that this was going to be recorded, so be careful what you say. But other than that, I hope that this will be really interactive. The person who should, among other reasons, because I think most of you know much more about some of these issues than I do, and in particular, the person who really knows what's going on here is a very talented grad student in the lab, Jerry, who actually did all of this work. So I brought him here to A, correct me when I say something that's wrong, and B, rescue me when you ask hard questions. So I'm going to talk about invasive physiological recordings from human patients with intractable epilepsy. I'm going to talk about data from 48 different patients. You can actually see the brains here. Each one of these dots corresponds to one electrode that was implanted in these patients. And these are patients who cannot be treated pharmacologically, and therefore, over the years, we've worked with an amazing set of neurosurgeons that implant electrodes inside the human brain to localize where the seizures are coming from, and also to functionally map what's happening in cortex in order to resect the epileptogenic areas and make sure that they are going to be able to get rid of the seizures while at the same time not impairing cognitive function at the same time. Roughly speaking, we've worked with two different types of electrodes. These are microwire electrodes, 40 microns in diameter, high impedance, that record data extracellularly, and I'm not going to talk about those electrodes at all today. Every now and then, when the stars are aligned in the right way and everything happens in the right way, we can get relatively nice action potential recordings from those microwires. And those are mostly localized in the hippocampus and medial temporal lobe structures. What I'm going to talk about today is what some people refer to as ECOG, or electrocorticographic recordings. These are 2 millimeters, low impedance intracranial field potential signals, and each one of those is one of those dots that you saw in the previous slide. Because the neurosurgeons don't know where the seizures are coming from, they put a lot of these different electrodes. I'll show you some numbers very soon. And they are distributed in many different parts of the subdural surface of the human brain. So these recordings give us millisecond or sub-millisecond temporal resolution. That we certainly don't record spikes. We record field potential signals, which we don't really understand the biophysics of very well, and capture. I think about these intracranial field potentials as an average of local field potentials. That doesn't mean that I understand local field potentials very well either. So this is an average of these local field potentials that people record from microwires. So most of what we've been doing is looking at invasive physiological data within particular tasks. But what I want to talk about today is the line of work that Jerry started, which has to do with combining data from these patients that we have over multiple days. The patients stay in the hospital for about 7 days or so, and try to look at interactions between brain areas. And so in terms of connections and interactions, there's a panoply of different nomenclatures and signals and resolutions that people use. So people talk about specifically neuronal connections at what I call the microscale. This is, for example, what Jeff Lichtman is doing with electron microscopy. What I'm going to talk about, and then there are people who look at macroscopic signals such as what people do at MGH with DTI. What I'm going to talk about is somewhere in between, on a scale of millimeters of cortex, which I refer to as mesoscopic. Another important distinction is between anatomical measurements and functional measurements. This is an anatomical signal. DTI, well, DTI is DTI, reflects the water flow. And what I'm going to talk about is physiological signal, so it's a functional measure of interaction between brain areas. We look at relatively fast signals over millisecond scales as opposed to neuroimaging correlations that people look at scales of seconds. And although most of my life I've been looking at transient signals over the course of several hundred milliseconds, today I'm going to talk about average signals over minutes, hours, and maybe even days of recordings. So let me just give you a quick flavor of what we've been doing for about a decade, and this is not what I'm going to talk about today. So we record from some of these patients, we present images, and then we record visually triggered signals, and we try to study questions about vision, about learning, about memory. And today what I want to talk about is this long scale 1 week recording. But before I do that, I just want to give you a glimpse of what these signals look like. So this is a case of one electrode in the inferior temporal gyrus in one of the patients. These are 39 different repetitions of that picture over there. Some of you may actually know this person. She used to work in building E25. Anyway, so these are presentations of this picture. This picture was presented for 150 milliseconds. Each of those gray traces corresponds to one trial. The green is the average of all of those trials. So you can see that there's a clear evoked signal in response to this picture. This is a study that Martin and Bill Lotter and Han-Ling Tang and many other people did, looking at what happens during pattern completion. I'm not going to talk about that at all today. These are signals that we can look at even in single trials. And again, the temporal scale of most of what we've been looking at is on the order of a couple of hundred milliseconds or so. So again, this is just a raster plot of this intracranial field potential signal, just to give you a glimpse of what happens on a scale of 100 milliseconds when you present a visual picture, which is something that we can reliably obtain from these type of recordings. So this is multiple different trials. Each one of these is a different picture. This is time in milliseconds. The color corresponds to to voltage, as indicated here. So what I want to talk about today is not this type of experiments. These type of experiments usually last about half an hour to 1 hour, depending on the patient and how willing the patient is to entertain us with our cognitive questions. But in addition to that, we could record data continuously. So we have 24 hours a day for about 7 days of data. And that's when Jerry came and asked, well, can I look at this continuous data and try to assess the extent to which different parts of the brain interact with each other? So I'm going to talk about data from 48 different subjects. This is the number of electrodes, and this is the number of days that these subjects stayed in the hospital. So just a couple of pre-processing. I want to go into some of the details of what we do with the data. Again, this is all pretty new to us, so I'm hoping to get feedback and maybe stimulate discussions, collaborations, and so on. So just in terms of how we pre-process this signal, we subsample the data to either 250 or 256 hertz. We use a notch filter at 60 hertz. Recordings in the clinic are significantly noisier than what some of you may be used to when you record from animal models inside a Faraday cage. We don't have that, so we need to have a notch filter at 60 hertz. We also have a notch filter at 20 hertz. Jerry discovered that there's some medical equipment has interference at 20 hertz as well. And the other thing that we do is that we use, we re-reference the signal to what we call a bipolar reference by subtracting nearby electrodes. And we think that this significantly reduces the spread of intracortical signals, but also helps us remove artifacts. Because we are going to be looking at a huge amount of data, tens and tens of electrodes over many, many days, over many subjects, and we're going to start looking at correlations between those signals. We were very preoccupied that we were going to be capturing artifactual correlations in the signal. So Jerry spent an enormous amount of time trying to come up with automatic ways of detecting artifacts and being very conservative to try to ensure that we're not, when we claim that there's an interaction between two areas or a correlation between two signals, that's not contaminated by different types of artifacts. So I already mentioned the bipolar referencing. We don't look at any pairs of electrodes that are less than 17 millimeters apart. That's because we are concerned about volume conduction. We don't want to correlate the signal with itself. And just empirically, we think that this is a reasonable threshold to ensure that we're actually looking at distinct signals and not signals from one electrode that are volume conducted onto the next. We remove clinically annotated seizures to avoid looking at epileptic seizures here. And then we have 3 different criteria to remove signals that are way too large for our taste, and we think that they're not really physiological. To remove cases where the derivative of the signal is very large, or also to remove cases where the amplitude of the signal is very small, and we think that those are cases where maybe the electrode is not making contact with the brain, or we're not recording real physiological signals. So after all of that, essentially we're removing about 2.3% of the data, and we think this is quite critical to try to ensure that we're not looking at artifactual correlations. So what I'm going to discuss today is we take signals from 2 electrodes, 2 bipolar electrodes, meaning after subtracting the neighboring one. So this is 10 seconds of data from one of the electrodes, 10 seconds data from the other electrode. And we're going to define putative interactions by computing the coherence between the 2 electrodes. So essentially, we compute the cross-spectral density, which is normalized by the auto-spectral density of the signals. This is a value that goes from 0 to 1, and it's computed at each frequency, at each interval t. So this is a segment of data of 10 seconds. We play with different values of t, and if people are interested, we can discuss t. And then here, people have used, as people know here very well, different Greek letters to connote different intervals over which one can do this type of analysis. And we have mostly focused on 2 for practical reasons. One is the broadband data, just integrating over the entire frequency interval. And the other one is by integrating this in the gamma band. Jerry has lots and lots of figures if people are interested in other frequency bands. And this is one of the many things that I'll be curious to get feedback and see whether we should or not expand the discussion. So I'm going to refer to these coherence values as mesoscopic, because we're talking about interference field potential, temporally average, because we're going to average over many of these windows of length t, functional as opposed to anatomical, putative, because we don't know whether these are direct interactions or indirect interactions, pairwise, because we're looking at 2 bipolar electrodes at a time, non-causal interactions in the broadband signals. This is a mouthful, so in order to avoid saying all of that all the time, I'm just going to talk about interactions. Yes?
Do you have some idea of the environment? Are they awake? Are they asleep? Do you have metaparameters for what signals are coming through?
So I'm going to be talking about right now the entire data set without consideration to all the amazing fundamental life of what happens. So we have video data. So in another set of ideas that we haven't, I'm not going to talk too much about today, Jerry, together with other people, have annotated behavior on a second-by-second basis. So yes, we have a lot of idea of what they're doing. I'm not going to talk about much of that today. So to begin with, we're just looking at these temporally averaged interactions. At the end of the day,
so you're temporally averaged over what time scale?
I'm going to show this so we use windows of 10 seconds, and we're going to average over a week of those. Okay, I'm going to show you what happens on each one of those 10-second windows. But at the end of the day, some of the numbers that we're going to give you are averages over many days of these signals. Now, ultimately, I think that a lot of the exciting interactions happen on a scale that's much shorter than that, on a scale of hundreds of milliseconds. And we have some tasks where we could look at those interactions, and we have looked at some of those shorter time scale interactions, and we have a lot more to do on that scale. So Jerry, I think, likes to call this housekeeping interactions. So it's just a sort of an average of interactions over these very long temporal scales. Okay.
Are you going to connect it to cognition, or do you think?
So not today.
Okay.
We have that. So if people are interested, so I think ultimately many of us are interested in what people are doing, whether people are asleep or they're not asleep, whether people are seeing, singing, talking. We have all of this data, and I think we need to connect it to all of these things. For today, I'm just going to take the entire data set and show you these average functional interactions. But I agree, these are quite fundamental issues. Okay. Okay. So when we start making this definition, the first question is, are we on the right path or not? And so we spend quite some time, and I'm going to spend quite some time today trying to give you some of the analysis that we've done to try to assess to what extent are we on the right path or not. And for that, we'd like to know. We look at data from humans. We're going to look at data from monkeys as well, because there are a lot of things that we know from interactions in monkeys that we just don't know in humans. And ideally, we'd like to have some positive controls, known interactions, and hopefully see whether we can detect those or not. Ideally, we'd like to have negative controls, areas that are not supposed to interact, and see whether our method detects that they don't interact. And then do the same thing in monkeys. And then transfer from monkeys to humans, because we know much more about interactions between brain areas in monkeys, and ask whether we can find those in the human brain as well. This has been much harder for us than we expected, in part because I think that basically we know nothing about mesoscopic interactions, and coming up with positive examples and negative examples, I think is quite challenging, particularly in the human brain, where a lot of data comes from non-invasive imaging studies where it's hard to compare. It's sort of an apples and bananas comparison.
So it would be easier in the macroscopic scale, you mean, for you, rather than the mesoscopic scale, to have some reference?
Well, we don't want to compare with functional neuroimaging studies, because I think that's a different beast. It's a completely different scale of operation. So we don't know whether we can use that as a control or not, because it's a completely different signal. So we'd like to come up with something that's at least somewhat more comparable to what we're doing. So I'm going to show you what we're going to be comparing against.
What about MEG data?
Right. So one could compare against MEG data. In that case, the spatial resolution is on a scale of centimeters, so it's very unclear to me, at least, and maybe you can correct me if I'm wrong, which brain area interacts with which brain area from MEG studies. I don't know. Maybe because it's very hard to know where the spatial resolution is. So we have a spatial resolution of millimeters here. So if we find an interaction, it's not very obvious to me. But again, these are kind of things that if people have strong ideas about how we can compare.
There's actually a study in 2016 with ECOG patients, and that was with 4 patients, and the question they asked was resting state interactivity, like functional connectivity. And the task was to sit still for an hour and a half, and that is just a different. It's just a different beast versus like when we're asleep, we're not really asleep, or when we're watching TV, we're not really conscious in the same way. So it's no seriously, like so watching TV is one of our behaviors. Just there's some preliminary stuff, it's pretty scary what you see when you're watching TV. So I think because you're in a hospital setting, it presents a unique challenge, but also opportunity to ask stuff about, like Monday, Tuesday, Wednesday, Thursday. These are the interactions that don't care what day of the week it is. And this is just like the best opportunity to ask that question.
Can I ask a little bit, for you to elaborate a little bit about using the bipolar electrodes and the subtraction? Like, do we do this in animal studies? If you have like an electrode surrounded by 8 others, why do you subtract 1 and not like all the 8 that are like, what's going on there?
So in animal studies that do this kind of ECOG analysis, people have also used this type of bipolar referencing. One major difference in many let's talk about single neuron, single electrode recordings. Monkeys or other species can be inside a Faraday cage, so you can reduce the amount of noise that you have a lot compared to what we have in the clinical setting. I don't have a good answer to why do bipolar referencing. So people have done what you mentioned. So some people call it a Laplacian reference. So you take one electron and you take the 4 values around that, and you subtract the average of those.
And there's common average referencing, right?
For a lot of our papers, we've done common average, which means subtract the average of the entire data set. I was very concerned that was not going to work here. I thought that, and we have some preliminary evidence suggesting that will introduce pathological correlations. So when you subtract the average of everything, and then you try to correlate A versus B, you may find a correlation that's because of that average that you're subtracting. So we think that subtracting a more local signal is much better than subtracting a common average for the purpose of looking at these interactions. Whether it should be bipolar or 2 electrodes or 4, my guess is that all of these would work to some extent. One of the appeals to us of only using 2 electrodes to define a signal is that then it's much easier for us to think about this interaction. So I can look at these 2 electrodes compared to these 2. If the signal from this electrode depends on 8 surrounding electrodes, then very quickly it gets very messy in terms of you may be correlating a signal that uses a similar one that subtracted a similar electrode, and so does that make sense? So I think it makes comparing electrodes, particularly because we look at everything that's more than 17 millimeters apart, I think it makes that calculation easier for me to understand.
Can you remind us what the channel count is? The total, because you were saying that CAR isn't a good way to go because it introduces spurious correlations. If you had a ton of channels, like let's say notionally, Dan will know what I'm talking about. Let's say you had 10,000 channels, then your concern with CAR would go away, right?
So in general, you can subtract anywhere from 0 up to the total number of channels. Our maximum is 186.
Okay.
The number that you choose is a balancing act between how much you can spare to lose and what kind of spread that you're aiming for. So we thought that by excluding diagonal neighbors and by subtracting direct neighbors, that was a good balance between not sort of killing our signal too much, but also not having extreme spread issues from not referencing at all.
So the average number is shown here. It's 86. That's the average across all the 48 patients. I think if you had 10,000, I would still be concerned. For example, let's say that the subject sneezes, and you sneeze, and the whole brain does something that will show up as an artifact in the common average. hopefully we get rid of those artifacts by Jerry's magical artifact rejection procedure. But I'm still a little bit, we've used the common average in a lot of cases where we look at single electrode data. In the case of looking at interactions, I'm still a bit concerned that the common average may introduce artifacts, even if you have 10,000 or a million. I'm happy to be proven wrong. In any case, the average number is 86.
Yeah, we have very similar a very similar situation to you where we have 60 hertz stuff that we can't get rid of, and I'm using a CAR approach right now, and I'm really intrigued by your doing the neighbor subtraction thing. I'm actually going to try it both ways and see which one works best for us.
So ideally, what you really want is to do a three-dimensional current source density analysis, which essentially amounts to subtracting a cage of 6 electrodes surrounding your test electrode from the potential at your test electrode. So that
in a perfect world.
In a perfect world, yes. So, you know, in the real world where you have strips of contacts and it's already one-dimensional, you know, I think what they're doing with the bipolar is probably the best you can get.
Well, so yeah, I also, so in addition to the bipolar, the common average, and the n nearest neighbor subtraction, I also looked at partial correlations and partial coherence. But the issue with ECOG is that it's not a whole brain recording, so there are potential unobservable latent variables that are affecting the data that I'm not accounting for statistically. But ideally, that would be, you know, the best, like taking like a simultaneous scalp EEG and ECOG and conditioning on that. But yeah, we just don't have that.
And I should say that Jerry looked at a lot of analysis with the common average as well a long time ago. And if you are interested, I'm happy to compare notes and exchange notes and tell you what we found there and compare with what you have as well.
That would be interesting. And ours is very spike centric. We actually filter out LFP. So I think that the best for us may not be the best for you.
Okay, so occasionally I step in as moderator and say, let's go on.
Yeah, we're done here.
Okay, so let me go through some of these preliminary attempts at validation. So the first one that we started with is what Jerry describes as perhaps one of the most ubiquitous description of an interaction in the human brain, which is the interaction between so-called Broca's area and Wernicke's area, two areas that are involved in language. And I mispronounce almost every word in English. I have a very thick accent. I'm not even going to try to pronounce the articulus. That's the stream of connections in the brain from Wernicke to Broca's area. So we found patients where we have electrodes that are presumably in these two areas, the left pars triangularis and the left inferior parietal cortex. These are the signals. We compute this coherence. We get a coherence value between these two for this particular 10-second segment of 0.34. These coherence signals are specific. It's not that every pair of electrodes that you can find in the brain will show you the same coherence. For example, during the same simultaneous 10-second segment, if you take the pars triangularis here and you correlate it with these other areas, you get lower coherence values. So this is our indication that for this particular, again, this is just a 10-second segment. I'll show you more data very, very soon. But we can find putative interaction in this case for two particular regions that do seem to communicate in the human brain.
Is this time dependent? If you pick the next 10 seconds, would you get something very different?
I'll show you that in a second. So to try to define a null hypothesis, what we do is we take one of the signals and then we randomly shift it in time. So in this case, we take the second signal, we shift it by 60 minutes, and then we correlate this signal with that one over here. This is what we call the null hypothesis. The coherence here is 0.21. We can compute the distribution of coherence values under this null hypothesis obtained by shifting the data. to try to come up with a threshold for what we will call a significant coherence or not. Okay, so next, again, it's very, very hard to get positive or negative controls from human data in terms of which parts of the human brain interact with other brains. So there has been much more work done in monkeys, trying to look at put electrodes in different areas and assessing whether they interact or not. So we moved on to monkeys to ask these questions. So for that purpose, and to try to compare apples to apples, or at least Gala apples to Macintosh apples, or some sort of more comparison. We used electrocorticographical recordings from monkeys. So we instead of spikes and LFPs, although I'm going to talk about those as well, but we used a data set from people in Japan where there are 128 ECOG electrodes, similar to the ones that we have, where they recorded continuously for 9 hours. And then we try to look in the literature for examples of pairwise interactions that people have described in monkeys. So this is one example where Bob Desimont's group looked at interactions defined by spike LFP coherence or LFP coherence between area V4 and area and the frontal eye field during a visual attention task. So these are two areas that are interacting according to this metric. So then we go into this ECOG data set, and then we find electrodes that are presumably in V4 and FEF. This is a completely different data set, and then we look at interactions defined between V4 and the frontal eye fields in the ECOG data specifically, and we take this as a positive control. So we do that.
Well, yeah, I'm a little confused because most of the tasks that Desimone and other people use go by very quickly.
Indeed.
So what are these 10 seconds recording?
Right. So the question is, with our definition of computing coherence in 10-second segments, and then in this case looking at 9 hours of data, can we detect interactions between two areas that are known to interact in those very short time scales? So we take that as an example of two brain areas that are known to interact. So presumably during those 9 hours, there may have been many segments, very brief segments where V4 and FEF interacted, and that's what we're hoping to capture here.
I see.
So in the human data, we're going to look at 24 hours. So we want to do the same thing with macaque data, but we want to have some sort of reassurance that...
So the monkey or person is doing many things during that 10 seconds or 24 hours, and you're averaging over all of the things that he or she is doing.
So I think this ECOG data, or this is 9 hours of spontaneous activity, or what exactly? I think this is 9 hours of spontaneous activity recorded with ECOG.
Yeah, it's 3 sessions of a 3-hour anesthesia task, where the first hour is rest, and then there's an hour of anesthesia, and there's a final hour of recovery. I cut out the anesthesia, so I just take rest. Yeah.
So this is 1 hour, 3 times 1 hour of rest. Is that correct?
3 times 2 hours.
3 times 2 hours of rest. Whatever rest is, which is somewhat comparable to what we're going to. Again, this is not perfectly comparable. This is we're trying to validate our metrics and see if we can. So out of 20 pairs of electrodes in these areas, we find significant interactions in 14 out of those 20. We do the same thing between V1 and V4 from data from this paper. And in this case, 116 out of 116 pairs do seem to show an interaction.
Sorry, was the post-anesthesia data also included?
Yeah, the post-anesthesia data was included. We can talk about that.
Yeah.
How long does it actually take for the anesthesia to wear off? That can be pretty significant, can't it? I mean, that can be hours.
It's a good question. I don't know. We probably I think maybe we should redo this only looking at the pre-anesthesia data. Maybe that's cleaner and easier and easier to interpret. Yeah. Yeah. So very hard to come up with negative controls. We'd like to have two brain areas that do not interact. Most people don't publish papers saying these two areas do not interact.
Are there any?
Well, how do we even know? So what we did was go back to anatomy and look at areas that are not anatomically connected as revealed by tracing studies. So this is a tracing study by Markov et al. By Kennedy's group, looking at different areas in macaque monkeys, and what you're seeing, the color here reflects a measure of how many axonal tracings they can find from one area to another area. For example, what is it? Area B1 has a lot of connections to area B2, as one might expect. So this is anatomy. This is not functional. So what we did was take an area here, the connection between B1 and probe M, and presumably there are no direct anatomical connections between these two areas. The fact that there are no anatomical connections does not necessarily imply that there are no functional interactions defined by our metric, but that's the best that we have been able to come up with as a negative control. So in this case, again, we do the same procedure. We look for electrodes in the ECOG data that are in these two areas, and we find that 7 out of 24 of the electrode pairs show an interaction. This is 0.29. It's not 0, but it's much less than what we found in the other cases where there is presumably an interaction. So next, we looked at the entire anatomical connections based on the Markov and Kennedy paper. Again, on this side you have anatomy. These are interactions defined by tracing studies in macaque monkeys. And these are functional interactions as defined by our metric with ECOG data from monkeys. So we see that there's a correlation between these two worlds. The correlation is 0.2. Not very impressive as a correlation metric, but again, bear in mind that these are completely different worlds, right? So this is tracing studies looking at whether there's a neuron that goes from area A to area B. The other one is a completely different monkey looking at these large-scale ECOG signals and our very coarse mesoscopic definitions of functional interaction. So I was surprised that we saw any correlation at all between this anatomical world and this functional.
What is the cluster in the previous slide? On the right side, there is the previous. Yes. On the right one, there is a cluster of areas in the center, or there. What are they? Oh, these are early visual areas. What are the other areas?
So this one, I'm more familiar with vision than anything else. So
Yeah.
this is B1, B2, B3, TO, B4. So this is the ventral visual cortex, more or less. Now this one, the 46 is frontal cortex.
Area 8 is the frontal eye field. It's the Brodmann, the macaque Brodmann, not to be confused with the human Brodmann. Area 8. So that's subdivided into LV. 9-46 are further subdivisions of related areas. 9, 10, and 12 are all like prefrontal areas, but these are clustered by distance.
So they are somewhere local in the brain, like around FEF, around.
These are ordered in a way that minimizes the distance between the neighbors, basically, so that what you're looking at should be relatively close.
I'm asking because I do expect some component of the interactions you will be detecting will be reflected in proximity, and then it's very hard to say because they're nearby or because when they're nearby they're also functionally connected. So that's a bit of it's hard to dissociate the two.
That's a good point. To make it even more complicated, we are actually excluding things that are very close by because we are concerned about volume conduction. So everything that's less than 17 millimeters apart, we're just removing those interactions completely. So that's.
So V1, V2, and V3, how can we get connections there?
In here?
Yes.
So I assume that these electrodes are more than 17 millimeters apart, right? Otherwise, we shouldn't be plotting them.
There are also more than one electrode on each area. But,
That might
yeah,
be
but
the furthest pair from between V1 and V3.
17 millimeters, is that a large distance?
no, so, okay, 17 so it's appropriately scaled for the monkey ECOG electrodes. The monkey ECOG electrodes are downscaled by a factor of 2, which is not exactly accurate because the cortical scaling is actually a factor of more of a 3 than a 2. So we just adjust that accordingly. But
Oh, I see. So 17 is for the human data, not for this. Okay.
But so for the distance question, even though we were excluding neighbors, that didn't stop us from asking the question of what does it look like as the distance increases, does the interactions fall off? And I don't want to spoil it because that's coming up. But in the Markov paper, they did find that there was a log normal relationship between distance and the log of the number of neurons that they found between each area, and that spanned an order of magnitude of 3. We didn't find anything like that drastic, but that's just a different technique. Yeah.
So my question is, everything you've been discussing, is like one-dimensional, saying the distance, 17 millimeters from the point of recording, but trying to visualize what other variable. I mean, it depends not just on the scale of distance, presumably, but in the relationship. Is it Roscoe, up, down, and have you examined that as a variable?
Yes, so the ECOG electrodes are titanium, where it makes the contact, but otherwise it's very pliable. It's encased in a silicone, kind of like an iPhone case. It's restrained in a certain way, since it is stuck, like it's physically constrained against the skull, to only record above the arachnoid membrane. So for that reason, it can't go into a sulcus. Now also, we have ground truth for neighboring electrodes. We know which electrodes are neighbors with each other. And we know which ones are diagonal neighbors from each other. And the distance that we use for thresholding is the distance that we get after reconstructing the CT and the MRI scans. So they kind of move a little bit. And then using the ground truth, we identified that 17 millimeters had a reasonable area under the rock curve in classification accuracy for diagonal neighbors. So really, it's a proxy for saying we're excluding diagonal neighbors since the electrode itself, the grid itself, is physically constrained.
Onward.
Okay. I'm going to go quickly. So we wanted to look at interactions from macaques in the human brain, and for that we needed to map macaque areas onto putative homolog areas in the human brain. Jerry did some magic that I don't quite understand completely to map one into the other. David Van Essen did some magic, which was re-implemented by Jerry. So now we can basically look at the same examples that I showed you before, now mapped onto the human brain, look at two putative areas, the putative homolog for area B4 and FEF, for example, and then we find that 14 out of 20 electrodes show a significant interaction. In the case of V1 and TEO that I showed you earlier, 100% of the cases showed an interaction where we were hoping to find an interaction. This is our negative control case, and out of 26 electrode pairs in humans, we found 0 that showed an interaction between area V1 and ProN. So then we looked at the map between the macaque neuroanatomical connections from Markov and Kennedy, and we compared that against the putative homolog areas in humans. The correlation is even weaker now. It's a very weak correlation, but it's still a significant correlation. Again, we're moving across millions of years of evolution from monkeys to humans, and from anatomy to physiology. So it's perhaps not too surprising that these apples and bananas comparisons are not that correlated. So to some extent, with a lot of caveats, I think that we try to validate some of these methodologies. So now let me move on with what we find with these measures of interaction. So Nancy asked earlier about what happens over time. I showed you one 10-second segment. If we look at other 10-second segments, there's a lot of variability, as one might expect. These correlations, these interactions change over time. So this is the segment I showed you before. If we move, I'm going to show you every single 10-second segment for one patient, but just randomly, if we move 70 hours earlier, there was a much stronger interaction. If you move 74 hours earlier, there's a little bit strong interaction, and here there's a weaker interaction. So there's a lot of variability in these interactions over different 10-second segments, as we might expect. Now I'm going to show you a whole hour. This is the point that I showed you before. So this is the coherence value. This is the 0.34 that I showed you earlier. And these are 60 minutes. Every single point corresponds to a different 10-second segment. As you can see, there's a lot of variability. Sometimes there are more interactions, sometimes there are less interactions. Again, not too surprising. Ultimately, we're interested in cognition to the extent that this is that has anything to do with the interaction between Broca's area and Wernicke's area, and to the extent that this has anything to do with language. One would like to know what's actually happening in these cases. We have those data. We could look at that. We haven't yet. We defined a consistency across time value, which is the number of points that are significant divided by the total. In this case, is 18%. So in short, there's a lot of variability over time, as one might expect. If we do that with the time-shifted data, essentially the CT value is very low, as one might expect as well. I showed you 1 hour. Now I'm going to show you within more than 140 hours. This is the entire stay from the patients in the hospital, this is the point that I showed you before. Again, this shows you a lot of the changes over time.
They're comparable with the null, right? So the point here is that there is no interaction over time, or because I see the null over there that you plot the small axis is 0.2 roughly as well, right?
This one and this one?
Yeah, what is the null over there? Is the time 60 data? I'm sorry, I'm missing.
The null hypothesis here is when we time shift the data.
Right.
That's right.
So the baseline interactions, if I were to call them, are comparable to the overall their values.
Most of the time, yes.
This, right?
Not all the time, but most of the time, yes. So most of the time, this is not different from this. But sometimes it's not. Sometimes there are...
So how I should interpret these ones is that there are, on occasion, there are a few spike or a few significant events, and now otherwise you go back to baseline, which is effectively truly null and not some baseline above zero interaction, right? It's really null.
Yeah, I agree. And the ratios in these two numbers
there.
What's that?
This is 0.002, and that's 0.2.
Yeah, so this is about 10 times larger than that. So that gives you more or less an idea of how many points there are here with respect to there.
Yeah, there are baseline shifts like you described. Unfortunately, there's no hard rules. It may vary within a patient and it may vary across patients, but sometimes there are large baseline shifts. Some of them I have looked at the video and they roughly correspond to epochs of awakeness or alertness. But the majority of the baseline shifts I have no clue about, and that's something that we're definitely interested in.
I am a bit confused. You plot and you say a number 0.002 over there, but if I can see the axis correctly, it's 0.2.
This is the temporal consistency. This is not there are two things. So one is how strong the interactions are. The other one is what fraction of all the time.
Okay, the spiky events.
Yeah, this is how many of these events there are. So this is a different metric. Sorry, this is how many time points are significant versus the number of time points that we calculate here.
So for the null hypothesis, you should really be controlling the null. So this should be a very small number, right? Because it's chance.
That's right. But every now and then, if you scrutinize this, I think there's one point that you will find some points. But that's
like the 0.05 control error rate or something. That's small. Yes.
So an important question, I think, it would be the calculations of the connectivity were done as an average of the entire time, or were you focusing on those moments in which something seemed to be happening compared to that null?
Yeah, we had been suggested to only take, like, let's say the activation as defined by some sigma over the mean. The final average coherence value will be the average of all of the points for which they're significant.
Okay, so you do focus on those kind of moments, instances in which there is exceeding. Okay.
That's going to be in the next couple of slides. So far, none of them has. I'm showing you all the data here. But now, if we look at these now, so now I show you one particular channel, one pair of channels. Now I'm going to show you one channel, one subject that is all the interactions from that particular area. The one I showed you before, I think, is one of these two. I don't know which one. One of these two. These are all the ones that were significant interactions doing this suprathreshold averaging for this particular set of for this one subject. So now, I'm going to look at all the 48 subjects, or at least all the subjects that have electrodes in this area. I'm sorry, I'm going a little bit quickly now. So these are all the electrodes in the pars triangularis, this Broca's area, and these are all the points that show significant interactions with this single one area. And in this case, there are 6 subjects that had electrodes in the pars triangularis. Just because I like the visual system, we had one subject that had electrodes in V1. These are the interactions from V1, which seem to make sense in terms of interaction with local visual areas along the ventral as well as dorsal visual stream. Someone was asking earlier about distance. So in some cases, the long distance interactions can also be strong. This is one example. This is the middle temporal area. I'm showing you all the interactions from all the patients that had electrodes in the middle temporal area. And in this case, you can see that there are many areas that are farther away. As a matter of fact, the ones that are farther away seem to have stronger interactions than some of the local ones for this particular location. So this is a summary of all the interactions that we have. This is now a grand average over all the different brain areas, all the different subjects, and all the time that the patients stay in the hospital. So this is the human brain interactome with all the caveats, all the different words that I mentioned at the beginning. This is the mesoscopic, putative, temporally averaged, functional.
So what's interesting is, I mean, many things interesting, but you can basically trace some lines there that are much, much darker than everything else. And what's the significance of that? So
let me first describe what's in here, and then I don't really know what the significance is. This is all new to me. First, the gray areas here are areas that are not covered. So here are locations where we did not have enough electrodes to actually make a statement. The white areas are areas that showed no significant interactions. Everything else, we find significant interactions. This is the value, the average value of the coherence between these areas. So, for example, the one that I've been talking about is the pars triangularis with inferior parietal, which is here, which is one of the strong but not the strongest interactions. There are several other cases that are that show stronger interactions.
What's on the right? Is that all of the electrode, all of the sites, or okay?
I think this is the rendering of each one of 5,000-ish electrodes.
Okay, so those lines are determined by the convolutional structure of the brain.
So the reason you see these lines is because of the convolutions of the brain and the way the electrodes are placed that are on the subdural surface, but they don't go into.
So the places where there are no dots are down in the sulci where you can't access.
Yeah, so
it's like a flattened brain view.
That's right. That's right. And just to verify, it's not that I've never done any surgeries. These are the neurosurgeons that do the surgery. So it's not that I cannot access. The neurosurgeons don't cannot access. I just want to clarify that. Yeah, but yes, that's right. So these are areas that.
So all the dots are on the surface.
That's right. Yes. So they're subdurally, but on the surface, and all of this is mapped onto a common brain. So there are distortions, and again, this is something that Jerry can discuss at length. There are distortions in this mapping from one brain to the average brain. So I think Jerry may
But
say that.
but the distortion is only a distortion when you visualize it, because the actual locations are anatomically defined by the curvature of each individual brain. So there's no really big erroring from like Talrock coordinates or.
But is it fair to say that, for
There
example,
appears to be some confusion about where these streaks are coming from.
Oh no, yeah,
If
the
the
streaks
brain's
are.
all folded up, and all these dots are uniformly distributed on the surface, and then you flatten the map, all the points on the surface are gonna lie along a particular.
Yeah, this is the central focus, just right here. Yeah, calibrant focus. Cocentral gyre. Precentral gyre. The width of this
absolutely
roughly.
nothing in the insula.
Yeah, nothing in the insula.
yeah, so on top of that, the neurosurgeons may not uniformly cover the entire. This is dictated by where they think seizures are coming from. So we have quite a lot of sampling over the brain, but it's not every single area is sampled. But you're absolutely right about the origin of these. Now, what I wanted to say is that, correct me if I'm wrong, but I think some of these points which seem to be outside of this may actually, in reality, they may actually be here. But because of this mapping and flattening from the individual brain onto the average brain, they are rendered here, but actually in that subject's brain, they should be in the same. Is that right?
Yeah.
So maybe it should even be more striking than what it seems here in terms of where the lectures are. Okay. So this is the summary of what we have. I'm not going to say much about significance. This was an exercise in characterizing who talks to whom in the human brain. What are the interactions in the human brain? I think we have a lot of work to do to connect this with cognition and with brain states and so on.
So can you describe at least I see one really bright feature there. What is that?
What is this yellow one here? Yeah. The one I told you about is this one here. The pars triangularis is this one. That's Wernicke's area to Broca's area. That presumably that's the interaction between the two most well-known areas in the human brain related to language. I don't know if you have a story about any of the others.
This is the story that I've been selling, or that we've been selling to you. But I mean, I'm really excited about this. So actually, I have several different versions of this, depending on how strict I enforce these parameters, because 2 is kind of just an arbitrary number. It's just to say that we're not basing it like just one person. Because, it's a patient. They have severe epilepsy. So it turns out if you jack this number up to 4 patients, that this region, so this is mostly due to the parahippocampal region, which is, it's still part of the cortex. It's a gyrus, but it's very near the hippocampus. It's not the hippocampus, but it's near the hippocampus. And these areas are roughly in the prefrontal cortex. So my original hypothesis was, I read about theta oscillations in the hippocampus from smart people like Matt Wilson. So I look at so I have this for theta. But the theta is not alone enough to explain the strength of this. So it's something that I'm still looking into. But I think, hopefully I'll be able to find something and try to talk about it.
But there's like a cluster there. Parahippocampus and what else?
So if you look along the axis, these are like neighboring interactions. So the more off diagonal you get,
The other one is, it looks like it's the pars triangularis. No, the other
So I think what they are
member of
asking
the
is,
pair
I
is the
think they
yellow
want you to
square.
read this. So this is parahippocampal. This is like right now.
Okay, all right.
I think.
Yeah, so these 3, the sandwich here is the parses. That's these 3 guys here. This is starting from the first bright line, that's the parahippocampal, which is right down here. It's just south of just below or wraps around the fusiform gyrus, which is at the very basal part of the cortex. The next one is the entorhinal area, so that's the neighboring one here. Temporal pole, medial orbitofrontal, lateral
frontal, and then the 3 parts to make the that moment here.
And just to be clear, this is symmetric.
Yeah,
Symmetric matrix.
this is painfully symmetric.
That's built into the math.
Yeah.
Yeah, sure. Question?
So maybe I very quickly want to say 3 things that of just future plans, and then I'll take this question. So that's mostly what I wanted to say. Just a couple of things. Once we have this, there's lots more that we want to do. One of them is we have basically a graph of interaction. So now you can throw in your favorite graph theory metrics. This is one that defines what's a small world network, and indeed it looks like this is a small world network according to the graph of different interactions. People mentioned before we really want to look at different states, what happens when subjects are asleep, what happens when subjects are awake. And ultimately also we have snippets of data where we have video behavioral annotation or where subjects are doing a particular task. So this is one of many tasks for which we have collected data where subjects are doing an association task between a picture, a visual stimulus and a response. And then we can look at a scale of 100 milliseconds or so, and more the things that many of us are most used to, which is looking at specific interactions that happen on very short temporal scales. So these are all in our to-do list. Maybe I think I'll stop here and just take questions and open it up for discussion. Yes.
Yes, have you looked at all at the spectral structure of these correlations?
Sorry,
The
The spectral structure of this correlation. So Jeremy has probably a lot. So he's looked at essentially all frequency bands. I showed you the broadband signals. We have all of the same plots in the gamma band, in the theta band, in the delta band, in the beta band, and so on.
I think they're secretly in this presentation.
They may be secretly in here. Okay, so
I'm the same for all those different areas. So the whatever the Wernicke's Broca thing and these other areas, is it the same kind of spectrum?
So this is one of the things that I don't know. I'm happy to hear. This is we can have everything here times 6 for each of these frequencies. The short answer is there's overlap across different frequencies, but they're not identical. So I don't know specifically about Broca and Wernicke's area, but if you look at that interaction matrix in different frequency bands, there's some overlap, but they're certainly not identical. There's variation from one frequency band to another. And this is something that we have tons of figures, and we're not exactly sure what to do with that. So some people favor one frequency band over another, or if there's a strong motivation.
Can I ask you what you found
yes.
Most surprising from this huge amount of data? What popped out at you as things you really didn't expect?
I think I don't know if I have an answer.
Whatever to the distance plot.
Well,
Okay, all right.
I'm not sure that's the most surprising, but anyway, to me, the most surprising thing is that we could find any interaction at all, according to this very coarse metric.
I see.
The fact that this is not all white. That's I have very low expectations. Maybe I'm a skeptic. I was expecting that we were not going to be able to see any interactions whatsoever, and some of them do seem to make sense with respect to some of the literature. But
so here's another point. You had some hours where you had rest and then anesthesia and then recovery from anesthesia. So there are theories about anesthesia in which the lack of consciousness is coming precisely from the lack of coordination in different parts of the brain. I mean, there are other theories that say there's too much coordination, and so that makes it impossible to have normal cognition. I think it could be interesting to use your techniques and compare the hour of anesthesia to the hour pre-anesthesia.
That was in the monkey data, though.
Yeah, we only.
yeah. So I agree. I think that I lumped all of that in the
Monkeys
brain
aren't
state
conscious?
but
No, the anesthesia data that they described was acquired in monkeys.
So what? Oh, you mean,
Both of you are right. That was reported in monkeys, and it's still an interesting question. One can still look at anesthesia or not. In the human brain, we could, but we haven't recorded data during anesthesia. Most of these patients only undergo anesthesia for a very short period of time during surgery. It turns out that you can do brain surgery without general anesthesia. If you've never thought about it, this sounds crazy, but you can actually, this is how many of these patients are actually operated. So yeah, we could, but we haven't. So all of these data are for monkeys, but you can still look at that. So one of the things we're doing is with the same in the same spirit, trying to look at sleep versus non-sleep. And Max Tegmark recently has been interested in these 5 related metrics of consciousness. We're trying to look at these as a way of looking at whether interactions do correlate with some of these different states. But in the monkey data, we can look at pre, post, during anesthesia as well. Yes.
Yeah, so ever since Li Wei Tsai gave a talk at the beginning of the year, there's been a lot of interest among this group in the 40 hertz oscillation band, because she makes a lot of interesting things happen simply by listening or flickers. So are you, with that data set, able to home in on potential events of interest or particular connections, you know, in that specific band? Because there are questions regarding significance of your data. I can see possible utility.
Yeah, we've looked at this in the same metrics in different in different frequency bands. So we have each one of these figures in different frequency bands. We haven't looked specifically at 40 hertz. We've looked at gamma from 30 to 100.
Yeah, so
but yes, we could look at if people are convinced that 40 hertz is where magic happens in cognition, we could look at 38 to 42. But
gamma is often thought to really be 2 different bands of a low gamma that's more like, 35 to 55 or something like that, and a higher gamma that's 60 to 90 or whatever. And they are probably doing very different things. So Li Wei has been looking much more at the lower gamma.
Yeah. So we probably should. So far we used 1 gamma band. We could only split this into 2 gamma bands. The more we split, the more figures we have.
It's fine. But just so that you originally, if you just survey the ECOG literature, it's a little bit of a controversial topic because there's work from Stanford by Kai Miller that sort of is showing that high gamma might just be an artifact of the electrical properties of the recording. So for that reason, we stuck to 30 to 100. But yeah, that could be.
Well, how high gamma?
Sorry?
I'm sorry. Some people consider high gamma way above 100. What
like a high frequency oscillation. I mean,
what are you talking about that may be an artifact of the recording procedure?
What frequency?
So we cap it at 100 because the Nyquist frequency for our recordings is 125.
Oh,
but
okay.
but high gamma can go as high as I think 200.
And it's probably
but you're
not
just talking
gamma.
about foldover problems, aliasing problems. Yeah.
The 125. Sorry, the 125 hertz is because of downsampling, right? The raw data has a higher sampling rate.
So we're putting together data here from different hospitals and different. So we have some cases as low as 250 hertz sampling rate, in some cases as high as 2,000 hertz. In most of the cases, you're right. This is because of subsampling. In a few patients, it's not subsampling. That's actually the frequency at which the sampling rate at which we record the data. But most of the time, you're right. It's due to subsampling. You're right.
Okay. So there's a really relevant preprint that came out last night on 100 to 300 hertz. They used stereo EEG to do really, really well localized and isolated in human epilepsy patients. And it's Gabriele Arnulfo is the first author. It's out of the Palva lab in Finland. And what they're finding is very significant, very long range, very high gamma interactions, and they think it's real. I mean, it's a preprint. It came out last night. I haven't read it word for word yet, but it really goes to exactly the stuff that we're trying to dig into right here.
Sounds interesting. We should look at that. Thanks.
Yeah, I'll get it to you.
Yeah, thank you. I think he was first. Sorry.
My question was about laterality. You presented these findings here. I wonder if they called in both hemispheres, or, for example, given the language localization, you may have found them only on the left side.
Is it fair to say that most of the data are in one hemisphere? We have less data in both hemispheres. Is that?
We don't overrepresent any right-handed people, but they are the way that we do these is we symmetricize everything.
So you didn't. I mean, I know like most are right-handed and the left language hemisphere, but I wonder then you could use it as a control or something if you don't find this interaction in the right hemisphere and you find it in the left, maybe you can associate it with some overall function.
Yeah, it's a good one. I don't know how many cases we have with electrodes in these two areas.
You are constrained with the limits, right? And you have tons of temporal electrodes because of the epilepsy.
Yeah, so for that specific interaction that we showed, there's 4 patients. 2 of them are right-handed, and then I think 2 of them have right-handed, right hemisphere coverage, but I forgot which one goes to which. So that'd be interesting.
Yes.
So I wonder if some of this high coherence between the parahippocampal and the frontal cortices, prefrontal, might be due to the fact that these are epileptic patients. Do you have a way of comparing it to? I mean, is there overconnectivity between those?
So this is it's illegal to study the brain of non-epileptic patients invasively.
But if you look at the monkey in the same area, do you think you'd see, or did you? Did you look at the monkey in the same area and see?
Well, the monkey wouldn't
Monkey actually has higher
Even
oscillation.
higher.
Yeah.
Oh, I see. Okay.
It's a little crazy,
wow.
but
Yeah, but in general, so what we do is we remove all the areas where there's all the time segments where there's a seizure. But at the end of the day, yes, you could say that the normal human brain is different. Just I don't know how we are ever going to study the normal human brain, and other than non-invasive techniques. But that's a completely different type of signal.
No, it seems like there's some evidence that.
But then in monkeys, maybe.
unfortunately, quite a number of these patients actually go through this and then have nothing out of it. Like just the surgery alone of implanting and then playing, because they also titrate the drugs while they're in the hospital, somehow stops their seizures, and so they just don't have a seizure anymore. That's why some of these patients can be in here for like 8 to 9 days. Because they're just waiting for a seizure to happen, and at some point they say, like, we just can't see here anymore.
Sorry, we cured your epilepsy.
Until we show you back on.
Until they go.
Have you tried making the same graph, but during seizure periods?
We have not. We have not. We could. I'm a little bit concerned that there may be more artifacts in that. I think some of the things that we call artifacts are actually seizures also.
Yeah.
That we're removing. But yeah, we could, and we haven't. I don't know who was first, but yeah.
It's exactly the same question as he asked. Have you tried computing this graph? But I will say during the anesthesia period.
That's what I was asking. I would really like to know if some of those connections go away or not.
You have 3 hours of these, right, I think?
Yeah, so
then you have slow wave sleep.
Yeah, so what we're mostly been looking at is sleep and non-sleep, but in the monkey data we have the anesthesia, we can look at that. We have the data, we can look at seizures as well, and yeah, these are good questions. We haven't, and we should.
So one major theory of anesthesia emphasizes connections between frontal cortices and parietal cortices. You know, exactly the kind of mesoscopic distances you're talking about. And so I think it would be very interesting to see if there are real differences in anesthesia. So what anesthesia is it? Because they're different and they have different...
Yeah, there is a drug. It's one drug.
Propofol?
I don't remember it. I don't remember.
Okay, because inhibition-based anesthetics like propofol can act really, really differently than, say, ketamine, NMDA-based, and may hugely change the kind of interactions that you're seeing. And in fact, it seems to me that anesthesia is the perfect test bed for looking at using the kinds of techniques you're talking about, because unlike normal cognition, which everything's changing on a millisecond time scale, with anesthesia, things are much more
Simpler.
They're simpler. They're not simple, but they're simpler. And so...
and it's human data, right? Did I understood well that the anesthesia data is human or monkey?
That's monkey data. So I don't disagree, and I think we should look at that. One of the reasons we wanted to look at sleep is because I think we have way more data to ask the same flavor of questions.
You could, except that in sleep there's also a lot of stages.
There are different
and a lot
stages,
of
that's right.
things that are changing, and
Right.
so.
Yeah.
Again, you're averaging over a lot of different, we can call them cognitive states that happen over a night's sleep, whereas in anesthesia, there really, as far as we know, aren't these different cognitive states. And it makes a lot of sense then to do this kind of averaging.
Yeah.
I mean, it would get at the differences between that and what you see in an awake situation, then gives you a sense of what happens between really no consciousness and behavior, at least on this temporally average scale.
You convince me. We'll do it. Oh, hopefully. Yeah.
So another pharmacology-related question. So these refractory epilepsy patients that you're testing, during the test period, are they on any anticonvulsant drugs?
Yeah.
And if so, all right. So, and if so, do you see any correlations between what drug is on board? Because that would be interesting to pharmaceutical companies. Some of these anticonvulsants, the real mechanism isn't pinned down, so it could be valuable.
It's a good question. We haven't looked at that. We do have, and at least in some cases, we do have data about which particular drug was administered and when. So we could go back to those plots that I showed you over 140 hours and say at which time points some of these drugs were. We have not looked at any of that. Yes, they are. They are under anticonvulsant drugs. They are titrated over the 5, 6, 7 days that the patients stay in the hospital. If they have a lot of seizures, then they get more anticonvulsant drugs. If they don't have seizures, they start to reduce them until they get no drugs whatsoever because they really want them to have seizures. That's the whole purpose of doing this procedure. So yes, these are all interesting questions. We have not done any of that.
Sounds like you have a gold mine of unmined information.
Yeah, we outlined a 20-year project for Jerry. Yes.
Do you guys consider doing any traveling wave analyses, or it seems to me with this much sampling, this much cortex, it'd be really interesting to see whether, I guess, establishing a little more directionality in these kinds of functional coupling, but especially with so much data, maybe establishing whether specific frequency bands tend to propagate in specific directions through our cortex, or I don't know.
Yeah, I think these are all interesting questions. The answer, we haven't done anything. Have we considered, have we fantasized about them? Yes. Have we done anything? No. So at least two things. One is these coherence metrics are symmetric. There's no causality here. We could look at causal interaction metrics of in pairwise interactions, which electrodes activated first. That's on our to-do list. The other one, which I think is you're also alluding to, is look at networks, not just 2 electrodes, but actually sort of larger ensembles with about 3 electrodes, 4 or 5, or even the whole network. These are all interesting questions. Jerry's amazing. They're only he needs to sleep too, but there's a lot to do, and we started with this as our simpler first step. But you're absolutely right. I think those are all very interesting questions.
Yeah, I think it also depends on how high the fruit is on the tree, because I think the most pressing question that I always get is behavior. Like you just left out half of neuroscience because you're not looking at behavior. So I think the first step before I'm allowed to do any motif analysis or any of the OLAP scoring stuff is to try to connect these things to behavior and see
But
how they
how can you
change.
do that on the time scales that you're considering? Because behavior is on the time scales of milliseconds to seconds.
Well, that was about 10 seconds, a phrase or an expression. So there are
So
some
then you
techniques.
would go back to
least-
thinking about
Yeah.
smaller time
At
scales?
least you can tell if a 10-second snippet came from an awake period or an
I
asleep
see.
period, and you
Okay.
can tell if it came from a watching TV period or a having a conversation period.
So we have some examples of specific behaviors where we have sub-second data during particular tasks. And then Jerry, together with other people, annotate for two patients, soon three, we have second-by-second behavioral annotations. These are undergrads that looked at the video. Anyway, so we have second.
project.
So, yeah, exactly. So, well, we like computer vision, but we thought it was going to be too inaccurate. So we have second-by-second behavioral annotations of are they talking, are they watching TV, and so on. So we could go that way. Yes.
Two thoughts. They might be related, but I'm not sure they are. One is, I know I asked about this before I was interested in the method. Now I'm interested in the interpretation about the binary or the electrodes, the subtracting the bipolar electrode. So if it works as a method for extracting or removing artifacts, that's cool. But when I'm trying to think of what the findings are, if I look at signal in an electrode here covariates with signal in an electrode here, that makes sense. Signal in an electrode here minus the average of the region correlates with signal in the electrode here minus the average of the region makes sense. The difference between two electrodes here covariates with the difference between two electrodes here is very what is the hand-wavy interpretation of what that is?
You can think of each electrode pair as being sort of like a crude version of a current source density. So that actually gets closer to what the cells are physiologically doing. They're issuing currents through their membranes in response to transmitters.
Okay.
So I think about it in exactly this way. But again, I think one of the main motivations for us to do this bipolar referencing is that I was always very preoccupied with artifacts. I was very preoccupied that the subject would sneeze, and I would be having a signal here, and I find a fantastic coherence of 0.8, and then it turns out that there's nothing there. It's just that these two electrodes were simultaneously at. So I think the bipolar referencing gets rid of a lot of concerns about global artifacts. What if the subject moved his head, or they scratched their head, or there's some other kind of artifact that we're not accounting for? We spend a lot of time trying to think about it, but at the end of the day, by doing this CSD-like, it's not really CSD, but by doing this, we get rid, I think, of a lot of
CSD.
What's that?
Poor man's CSD.
Poor man's CSD. I think we have a much higher confidence in getting rid of a lot of many potential confounds about these interactions. But
that's totally fair. And then the other thing is, again, sort of going back to, I don't want to say behavior, so I'll say function. But one thing sort of that is surprising but not surprising is that if these anatomical divisions between regions made a lot of sense functionally, that you would maybe expect a very strong diagonal. And the fact that you don't see a very strong diagonal might mean that these regions are functionally heterogeneous. And the reason I'm very interested is because, as you said, Roca and Muriki, Anya and I smiled because a lot of the work in our lab is you can't call Roca's region because there's a domain general part there, and there's a language selective part there, and in every subject they're located differently, so you can never know where they are without function. The same with Wernicke, it's like right by a domain general region that's in the inferior parietal lobe. Like these regions are so functionally heterogeneous and show such high inter-individual variability, at least, you know, at the level of fMRI, that like maybe, I mean, I'm not seeing the diagonal and I'm kind of weirded out by that.
So there's 36 data points here. Using a Wilcoxon rank sum statistic, it's not significant compared to the rest of the diagonal. Now, it's a miracle that I'm even showing diagonals, because in order for the diagonals
Right, they need to be far apart within the same region.
So, for example, precentral being a giant region, it has the privilege of having electrodes that cover within these regions. So actually, the regions themselves in this particular scheme that we use to parselate the brain is based on anatomy only.
Right.
So it's not guaranteed at all for anything to function. So the fact that this is not all great is already, you know, like something that it was extra because we basically were trying to kill these local interactions, because unfortunately, just because of the limitations of our understanding of the current source, we can't compare.
Do you often have electrodes in like similar regions across hemispheres, like homologs? Because that's one case when you might expect to see relatively high coherence as a sounding exchange.
So
maybe, I don't know, I mean, at least, you know, comparing it to fMRI, which is a completely different story.
No,
but
in the DTI world, there is strong cross-hemispheric interactions. We have two subjects for which we have both left and right coverage, and we also actually have one subject who came in twice, where the first time they left without a resection, and so they came back with healthy cortical tissue, and there was a overlapping, so there was a new electrode coverage, but some of the electrodes overlapped, and that is not something that we've looked at yet, but potentially we could look at how consistent it is.
So it's 6 or after 6.
Okay.
I'm going to suggest we thank the speakers. More is very welcome to stay and discuss more, and those who don't can politely escape.
Thank you, everyone, for all the feedback. This was very useful to us, so thanks for all the feedback.
I like those right left. I never thought of
Yeah. Thanks a lot.
For what it's worth about different frequency bands, the 40 hertz thing is of great importance. I don't know if you've seen this story, but for Alzheimer's and activating glia to clean up the beta amyloid mets. So they found that they can actually reverse the formation of amyloid plaques in Alzheimer model mice by subjecting them to 40 hertz flickers or 40 hertz click trains.
Yeah.
And then
I'm happy. I mean, we can. Part of I'm happy to look at one specific frequency band. We could.
My personal favorite band is beta, especially low beta. So it's another one of those two-half bands.
So Jerry looked at all of these, and we have. It's just that. In some sense, we have too many figures and too much, and we don't know exactly what to make with all of. It's not that
It's
beta.
good that you presented like a small coherence instead of tons of.
Well, it's not obvious to us, at least in what we've done so far, that there's one frequency where it's completely different from everything else. At least at this scale, in this context, and so on. Now, I think when we say my favorite band is beta, I think what you mean is in the context of motor control or the different signals. When I look at these
just in the context of large-scale networks.
Okay.
Ephemeral large-scale networks.
Yes. Okay.
Always seem to have something to do with beta.
Okay. I'm happy to send you all the.
My favorite is theta, actually.
Well.
No, it is.
But it's not always.
So then theta or delta are kind of like an organizing rhythm within which you have beta. I mean, all this nesting stuff actually.
It's not always to us that we see anything completely different, I would say, or is that a fair representation that it's not that one band we see everything and the other one we don't, for example.
Right.
We don't have
But you're not looking at the behavior yet. That's the whole, I mean, if you want to get into cognition, that's only going to come from correlating the behavior with.
And maybe when we do that, then we will, absolutely. I agree. At this scale, with the kind of things that we're doing, we have similar but not identical plots for
Yeah.
each frequency band, and that's where we are right now.
Yeah. This is
So, hey Dan, can I jump in?
Pairing this with anatomical connectivity studies is an excellent place to start. So, bravo.
I just wanted to ask, I promised Nancy you're right, so just
Yeah,
quickly,
okay.
I brought up 40 hertz because actually 2 weeks ago I gave a presentation about a new theory that might explain how these interactions occur where you get flickers or auditory stimulation producing microglial activation. And it involves thinking of 40 hertz frequency band as a carrier wave for information. So in listening to your talk, I got kind of excited about the possibilities that examination of your data might provide some degree of testing of that hypothesis. So I just wanted to get it on your radar screen.
I'm happy, I forgot to
So
say.
like where the tuning fork is 440? Yeah, 440 times.
No.
I haven't thought about that.
Yeah, 40 and beta.
If you want, for some context, basso profundo, Tibetan monks, alpenhorns, they're all at 40 hertz.
So I know you have to go, but send an email and I'm happy to continue.
You're writing something
Like
up.
I ask you something
Yes.
from my personal perspective. I don't see directly the connection here unless there's some other kind of data that you do you see any connection with this thing with mesoscopic measurements? How would this inform somehow what we can capture from outside? I didn't, no, I know, I also didn't see directly, but do you have any data with images or something?
We have simultaneous EEG, scalp EEG,
Oh.
or a small, like I can count on my hands.
Okay.
And it's not a full 10, 20.
No, but most of the cases we don't have any other.
And I saw like you had a slide with some cognitive experiments and images. Do you have this data or like what was
With the cognitive experiments, yes, we do have several of these experiments.
with
and we have-
patients or
with these patients, yes. So that's the way in which we
So
can
when
look
you
at.
like vision, I don't know whether at some point we could talk about linking, like I'm running the neck facility here, maybe we could think of something common together in this aspect. So no brain rhythms here, but
Yeah, absolutely. Send me an email. It's hard for me to understand how, as you know very well, better than I do, that there's a whole industry of fMRI people looking at resting state connectivity, a whole industry of DTI people looking at the connectome, right? I don't know how to link this to that. Maybe there should be some relationship, but I don't know exactly what
my question
that.
was not directly on resting state and connectivity, but I thought that you have some
On vision,
like side data with the cognitive task and images or something,
absolutely.
or in general. I mean, in general, talking to you, like.
I'm happy to talk more. Send me an email.
Nearby,
I'm happy to chat more. We can grab lunch and whatnot. And it's just these are very different signals. I'm not exactly sure how to compare them.
I'm not sure, but I think there should be out there some monkey papers looking at resting state fMRI and recordings to try to figure out what and whether there is a relation. I mean, I'm sure someone did this, and I can't recall.
Yeah, they did. They've done like a lot of comparisons. Yeah, and higher than this, definitely.
And the other place is where is the lab that Ev keeps talking about? There is one lab somewhere. In Iowa, yeah, and they scan people with electrodes, with grid electrodes.
Okay.
So they can do the same thing in humans and try to see whether, what you record from the electrodes and the magnet even is, yeah.
Yeah. I
don't know if they have anything out.
So you have a plan to collect even more data, right? This is an ongoing collaboration, I assume, or you want to expand this to more and more?
We have so for this business, this is actually a lot of patients.
How many?
This was 48.
48.
We have most people publish papers with 10 subjects in this domain. So I think this is actually
no. I misunderstood.
We hope to collect
I also
more.
kind of got mixed in my brain. Like there was at some point there was a monkey talk and then human. I kind of mixed it together. I don't know what is what data.
The reason we went into monkeys is because
I was surprised when you told me about like the anesthesia and then it was a monkey.
Sorry, that was confusing.
I got confused. I mean, it's fine.
I think I managed to
I
confuse
figured it
everyone.
out maybe like.
No, I think I managed to confuse everyone.
Yeah, I sort of got the impression the anesthesia was in humans to start with, too.
No, yeah, okay.
But that was kind of an offhanded comment.
Yeah, no, I should have, I think I managed to confuse everyone. You should have given the talk. He's much better than I am at giving the talk, so.
But the data collection is like relatively low effort, right? The patients will already coming in, you're not like filtering them for anything, just after the recording anyways. Is that right?
The tasks are huge effort.
Right.
So when we actually do
most
that,
of what you're looking at.
but this sort of continuous data that happens whether we exist or not. That's right. Yes. Yeah. Mm-hmm.
Well, they pay for it, so that, because it's part of treatment. This is their medical treatment.
So like if you wanted to double the data set, like how long would it take?
This is a decade, so double this data set is not
you can double this tomorrow if you want.
White hat.
What?
we have a lot of unprocessed data.
Oh, nice.
So it's just kind of.
No, this looks
no,
like
but if you want to collect data from
amazing
50 patients, and unless you, we're starting the collaboration with people in China. Maybe in China you can do it much faster. Well, they have lots of people there.
But in Cleveland Clinic, for example, like they're having tens of patients every week. They have.
Is that so? Okay.
Yes, a lot. Like it's the biggest epilepsy clinic in the U.S. and John Moser was there. Like I know him, a personal friend, and now he moved to Houston. Like they have really a lot of patients.
The volume depends a lot from one hospital to another and so on. So
like I remember there was a factory there having patients in rooms. I went and visited, like in a few rooms they had patients, sitting in bed and recording continuously EEG with.
Well, no, but there's a difference between EEG. No, EEG happens. No.
Subset of them, indeed, you're true. Subset of them.
If you want to scalp EEG, that's very easy, very cheap. You can get lots of that.
But subset of them go with invasive. That is true. Like.
Right. No, so we have also lots of patients. We haven't looked at those, but we have lots of patients with EEG. So, but in invasive data, I think it's less.
And then if they have behavioral tagging for all 48, or like what?
No, we have behavioral tagging for
two patients, 24 hours each.
And those done manually?
The third one is halfway done. Yeah.
So manually, like.
Or MTurk. You could get a lot done. But that would cost a lot more money.
So we didn't want to do it on MTurk because these are patients and we don't want to. So we did all of this in house, and so we didn't want to do that.
No, that's right.
So we have.
There's like they publish like Nature papers on doing like long-term multi-hour recordings of fruit flies, and then they do like clustering on it, and then they tag the behaviors post-hoc. I haven't looked into that at all yet, but we could potentially use like the 24-hour annotations that we have, and then try to cluster it based on that, and then try and see if we can try to label some. But I don't know.
So right now what we have concretely is two patients, second resolution behavioral annotations.
24 hours is a lot.
Yeah, it takes a long.
Well, yeah.
Although you can get through the sleeping periods on fast forward.
Well, yes.
The undergrad students love meditation.
Well, no, it's a list of 10 behaviors, and so you click it when it's on, and then you click it when it's on. So it's not like a.
And you have
strictly speaking,
independent
you have
observers to confirm.
So we did. So not every. Yeah, not everything, but some parts we have 2 observers, some parts we have 3 observers, and they are they are quite consistent. They're not perfectly consistent, but they're quite consistent. When we say somebody's watching TV, most of the time that's but you're right. So they're not 100% consistent, and
well, that's human.
and undergrads are funny people. But yeah.
I need to go, but thank you so much.
Yeah. All right. Thanks a lot for the feedback.
Hey, Jerry.
Yes. Hi,
You said, yeah, this is Daniel. Yeah. You said that, here, I can actually turn my camera on. Hang on a second here.
Okay.
You were, there we go.
Okay.
You said that you had some citations on what's going on inside people's brains functionally when they're watching TV, and that it was quite alarming. And I sent you an email. If you could reply to my email with 1 or 2 of those sites about watching TV, I really want to see that. That sounds like fun.
Wait, no, oh, not citations. Like, I like briefly looked into TV watching, and it was just that it's just like the number of significant interactions that we find drops. Like it's visually very.
I was going to ask whether it looks like anesthesia.
Not in the visual cortex, it doesn't.
We don't have anesthesia in humans, unfortunately.
But you can tell whether TV watching relative to non-TV watching looks like anesthesia relative to waking in monkeys.
Yeah, I guess so. We could do the interspecies.
So I would guess what you see is a collapse of functional connectivity and motor-oriented stuff.
So the issue with this is that each patient that we have these videos from only has like a limited scope of coverage for that patient. So the power only comes from combining multiple different patients. This one in particular did have like 50% of the parietal lobe, which is presumably motor and somatosensory related. So I didn't look into this like too deeply, but yeah.
I guess it's not a big surprise.
Yeah.
There, my video came live.
Yeah,
It did.
live.
Finally.
Are those meaty cables in the back? Is that, or are those
I'm sitting in a microwave engineering laboratory. The cables that are hanging there, those are all 26 gigahertz. Yeah, we're talking about 100 hertz here, 125 hertz. There's a 13 gigahertz oscilloscope right here. Got some crazy gear in here. Yeah, I fired you guys an email with the site about the high gamma stuff.
Yeah. Wait, those aren't the high frequency oscillations, are they? Like the ones that they use for I think they're using it as justification to put like specific depth electrodes that also are like there's some special depth electrodes that they're putting in.
No, this is
up to like 1,000, they say.
This is no, they top out at 300 according to the abstract and some of the body which I read. It's stereo EEG, so they're just doing like fancy localization.
This is stereotactic, like SEEG.
No, not stereo. It's SEEG, which I mean, I'm not an EEG person, so this is kind of new to me. But apparently, SEEG is just a way of like they get submillimetric accuracy in the localization of the source that the EEG is picking up. That's what they claim, anyway.
Wait, this is with the robot, like with the. Do they have like a metal frame?
I believe it's non-invasive. It's in humans, by the way. This is a human.
Okay, cool.
Keep in mind, you can't, mathematically, you can't have more independent points in your reconstruction than you have EEG channels in your data set. So.
Indeed. Valid point.
They're actually, they claim that in fMRI they can go up to gamma now. I went to one of the job interviews for the Department of Psychology at Harvard.
Mm-hmm.
And the applicant was claiming that they could observe gamma oscillations in fMRI.
Well,
wait. They're saying the bold signal, because that's about the bold signal. Are they saying that the bold signal is...
It's some kind of...
I mean, you're talking like 80 dB of loss. I mean, it's just a crazy amount of loss.
Well, you just invert the frequency response of the bold signal, and then you have unlimited bandwidth.
I don't know. The gist of it was...
Unlimited noise.
Yes.
That's very intriguing. I mean, if that's right, if they have like 2 to 3 cubic millimeter localization up to low gamma using the bold signal, that itself is some real... Because there's a whole bunch of stuff we want to look at that's, subcortical. We want to look at thalamus and basal ganglia, and it's just such a pain to get to it.
Yeah, we have some depth electrodes, but there are not enough and consistent enough that I mean, there's still like a lot. It's just we just haven't looked at it yet. But they can hit anywhere from basal ganglia to... They're mostly targeted towards like cortical areas,
Mhm.
insula, but yeah.
Yeah. Two weeks ago, Dan presented a paper in this group here, and it was a
Thank you.
Yeah.
Great to have you here.
it was a human study, but they could only look at the STN because it was Parkinson's patients, and you can only look at what is medically necessary to put electrodes in. So they had spike data, but it was just in the STN. Subthalamic nucleus.
Oh.
Because for like the last 15, 20.
It's a little nucleus that's like underneath the striatum, and logically, of course, also underneath the thalamus. And it's the most frequently targeted place for deep brain stimulation treatment of Parkinson's.
Right. So that's why they have electrodes there. It's just because they're doing the, they fire. You know what working frequencies? It's 150 hertz or something, and the DBS stuff. Anyway, they basically like knock those cells out by just continually bombarding them in the STN, and it's a long story, but it would be absolutely brilliant to have 40, 50 hertz functional response volumetrically instead of just on the surface.
I got a friend from undergrad who's going to UCSF for his MD PhD, and his life dream is to be able to do human optogenetics with deep brain stimulation.
So,
good luck with that.
Right. We see these. We have competitors. So my day job is actually doing a brain implant. We're doing engineering for hyperscale brain implants. And we keep it straight ahead. We're just actually using microwires. There's no genetic engineering, no germline, no craziness. But we actually have competitors that say it's targeted for humans. Yeah. We're starting with sheep in I think a month or two is when we're going to first. So we do it's 64 kilochannels with a 32 kilohertz sample rate. So it's truly hyperscale. Yeah, we run our payload is more than 3 gigabytes per second.
Huh. It's loading point precision. What?
It's 12. It's just 12-bit samples at 32 kilohertz. So it's all like but we just look at spike data. It's capacitively coupled at the head end. So we're just looking at spike data. So we actually need to have the high sample rate in order to be able to see the spikes. And potentially do some spike sorting. But spike sorting on 65,536 channels is computationally challenging.
Yeah, well, it actually, believe it or not, practically speaking, it gets a lot easier as you have more channels.
Yeah, we talked about that, Dan, and I really appreciate how at least we don't have to do these full really hard to grasp sort of fitting algorithms and such. Instead, it's just you've got one close to it, you've got one further away, and there's one other one that's even further away from it, and you're completely surrounding every unit. So it just becomes a lot more straightforward.
Yeah.
At least I don't want to put in the cart ahead of the horse. We haven't done our in vivo with hyperscale yet at all. But it's coming soon.
So it's down to just the three of us now. So I think we should wrap up the session.
I think so.
Let's see, who else? Nope, it's just us chickens.
Yeah. Hey, thanks a lot, guys. That was tremendously. You guys just saved me like 6 hours of coding. I do all my coding in C, so it hurts.
How did they save you coding in C?
Because I was about to go implement CAR for our live channel. So what I have to do, I have to actually cure. We have so many channels that I have to have an automated way of throwing out the channels that don't have good data in them, and that involves something. Just see, is there spiking data in the channel at all? And the way I'm going to do that is with like a peak and hold thing, but I have to condition the signal before I can do a peak hold.
You definitely want to use channels that are not necessarily adjacent, but at least in the same neighborhood. I'm just going to do the 8. So we get the data in. We actually use our technology is derived from imaging technology, like actual focal plane array imaging.
technology. So we have a very natural spatial surround kind of thing, and we actually press a bundle that's been nanoscopically smoothed and to make even contact against our focal plane array of conductive contacts. So all I'm going to do is I'm just going to subtract the average of the 8 surrounding pixels. We call them pixels from the channel, and I'll be done. I mean, that's just,
that should be perfect. Yeah.
Oh yeah, well yeah, make sure to average it. But yeah.
Yeah.
yeah, I won't be
He's
average.
doing the current source density thing.
Yeah.
In his case, it's
but it's plain. I mean, it's planar.
Yeah.
But I mean, it's going to
work.
work.
Them too. It's just that we have 1 by 8 strips. So like, what are you gonna do with a 1 by 8 strip? Like that doesn't make any sense.
I think just bipolar is a much better way to go with that.
But clinically, they use both CAR and
yeah.
So this was I got a triple whammy. I'd say I got a triple whammy out of your guys' talk today, because it's like day job. And if you want, I can tell. I'm doing a talk next week on my work. I moonlight as a theoretical neuroscientist. So the long range functional connectivity stuff is way up my alley. Also, the anesthesia experiments. That was Nancy that was pitching the anesthesia experiments, right?
Yeah.
So I'm really, really interested in that, and if you're not already spun up on what sort of shifts you see in directional functional connectivity as a function of anesthesia, I did a deep dive on that about 1.5 years ago, which I kept up to date with more recent literature. So I mean, I have a very compact little thing. I could just blurb you that if you want me to get you rolling, if you don't already know it all.
No, I don't know anything about any of this. I know that it makes waves or something, right?
There's two phenomena. You get a collapse of functional connectivity in some bands, but you also get this like macro scale synchronized oscillation. Exactly the type of thing that you would like subtract out if you were doing CAR.
Right.
And an LFP context.
Like kind of like a heart propagating.
Like a what?
Yeah. Like a heart. Because the heart kind of.
It does look like some of the physiological data that you want to subtract out. That's true. Your email address.
Oh yeah.
I took a guess at it. Was it? Got a frog in my throat. Is it Jerry.Wong at tch.harvard.edu?
No. It's I'm here. Yes, I'm with the GSAT school. So it's jwang04 at g.harvard.edu.
I wonder who I sent an email to about half an hour ago.
I don't know. Maybe they'll find me and like.
I'll send you a copy. I just sent the pointer to the high gamma thing. All right, let's fail. We've all been here for.
Yeah.
I've been here for 2.5 hours. All right, thank you very much.
Awesome.
This has been,
thanks.
been great.
Thanks for
right.
Thanks for the feedback.
Okay. See you
I'm
soon.
hanging up. Yep.
Yep.